Semi-conservative replication means that each strand of the parental DNA acts as a template for a new strand, which is added by base pairing. The parental DNA is labeled and then allowed to replicate to cells for a new generation, as the new DNA they make are unlabeled. DNA replication happens in two general steps. First, the double helix is unwound to separate into two template strands and make them available for new base pairings. As the nucleotides form complementary base pairs with template DNA, they are covalently linked together by phosphodiester bonds, forming a polymer whose base sequence is complementary to the bases in the template strand. The template DNA is read in the 3 to 5 direction. During DNA synthesis, nucleotides are added to the 3 end of the growing new strand. DNA polymerases add nucleotides to the growing chain. DNA replication begins with the binding of a large protein complex to a specific site on the DNA molecule. This complex contains DNA polymerase, an enzyme which catalyzes the addition of nucleotides as the new DNA chain grows. All chromosomes have at least one region called the origin of replication, or the ORI, to which pre-replication complex binds. Binding occurs when proteins in the complex recognize specific DNA sequences within the ORI. In prokaryotic organisms, the ORI sequence is at a fixed location on the chromosome. Once its pre-replication binds to it, the DNA unwinds and the replication proceeds in both directions around the circle, forming 2 replication forks. The ORI sequence binds at the replication complex, and the 2 replication forks grow away from one another. In eukaryotic organisms, they are longer than prokaryotes, and they are linear, not circular. There are multiple origins of replication, and the replication forks move away from each other. DNA replication begins with a primer. DNA polymerase elongates a polynucleotide strand by covalently linking new nucleotides to a pre-existing strand, but cannot start without a short starter strand called a primer. In most organisms, the primer is a short single strand of RNA. In others, it's not RNA, it's DNA. Primers are complementary to the DNA template and are synthesized one nucleotide at a time by the enzyme primase. DNA polymerase then adds nucleotides to the 3 end of the primer and continues until the replication of that section of the DNA has been completed. Then, the RNA primer is degraded, DNA is added in its place, and the resulting DNA fragments are connected by the action of their enzymes. When DNA replication is complete, each new strand consists of only DNA. The 2 DNA strands grow differently at the replication fork. A single replication fork opens in one direction. Imagine that DNA opens from one end like a zipper, and remember that the 2 DNA strands are anti-parallel. The 3 end of one strand is paired with the 5 end of the other. DNA replicates from the 5 to 3 direction. One newly synthesized strand, the leading strand, is oriented so that it can grow continuously at its 3 end as the fork opens up. The other strand, the lagging strand, must be synthesized differently because it grows in the direction away from the replication fork. The synthesis of the lagging strand requires the synthesis of relatively short, discontinuous stretches of the sequence, called Okazaki fragments. These are synthesized just as the leading strand is, by the addition of new nucleotides one at a time to the 3 end, but the new strand grows away from the replication fork. While the leading strand grows continuously forward, the lagging strand grows in shorter backward stretches with gaps in between them. A single primer is needed to initiate the synthesis of the leading strand. Each Okazaki fragment requires its own primer to be synthesized by primase. DNA polymerase then synthesizes an Okazaki fragment by adding nucleotides to one primer until it reaches the primer of the previous fragment. Now, a different DNA polymerase removes the old primer and replaces it with DNA, and a tiny nick is left behind. The final phosphodiester linkage between the adjacent Okazaki fragments is missing. The enzyme DNA ligase catalyzes the formation of that bond, linking the fragments and making the lagging strand whole. DNA replication occurs at a rapid speed and is extremely accurate. When the terminal RNA primer is removed from the replicating end of the linear eukaryotic chromosome, no DNA can be synthesized to replace it because there is no 3' end to extend. So, the new chromosome has a bit of single-stranded DNA at each end, which activates a mechanism for cutting off the single-stranded region, along with some of the intact double-stranded DNA. Thus, the chromosome becomes slightly shorter with each cell division. Chromosome ends must be protected from being joined to other chromosomes by the DNA repair system. When DNA is damaged by external or internal agents, it is repaired by a combination of DNA polymerase and DNA ligase activities. This system might mistakenly recognize chromosome ends as breaks and join two chromosomes together, creating havoc with genomic integrity. To prevent chromosomes from joining, many eukaryotes have strings of repetitive sequences at the end of their chromosomes called telomeres. These repeats bind special proteins that protect the ends from being joined together by a DNA repair system. Also, the repeats form loops that have a similar protective role. Telomeres act like plastic tips of shoelaces to prevent fraying. Apoptosis, programmed cell death, explains in part why many cell lineages do not last the entire lifetime of the organisms, because their telomeres are lost. Enzyme telomerase catalyzes the addition of any lost telomeric sequences in continuously dividing cells that maintain their telomeric DNA. Telomerase contains an RNA sequence that acts as a template for the telomeric DNA sequences. Relationship between telomere length and aging is that the average telomere length is shorter in older individuals. Errors in DNA replication can be repaired through proofreading and mismatch repair.